You ready?
Ready. Hello, everyone. Welcome to our final webinar for 2014. Carol and I will provide you with a year-end summary of some of the very exciting research that's been going on for 2014 and a little bit before, as well as an update on a lot of the federal activities that we try very hard to keep our finger on the pulse of. So let me turn the webinar for a moment over to Carol Head, our President and CEO, so that you can hear her voice and so she can give you a little bit of an introduction. Carol.
Great. Thank you so much, Suzanne. And welcome to everyone who is listening to the webinar. We thank you for joining us and being part of our community. It's been an interesting year of change for us here and change and advances in the illness as well. And much of what's occurred in 2015. We are cautiously hopeful that 2015 can be a tipping point year for this devastating and neglected illness. I say that for a couple of reasons. First, the federal attention to our illness has been much more pronounced than in prior years, and for good or bad, and we are cautiously optimistic that it will be good. The federal attention shines a bright public light on the scarcity of attention and fundraising to our illness. And second, there are a number of new research efforts that are commencing now and in the coming year, both ours and others. And again, we are very optimistic about new efforts with research. Meanwhile, today, we asked Suzanne to bring some order to our minds about the research that's occurred in 2014 regarding this very complex illness. If you're like me and many people, it's very difficult to organize the research in my mind. Those of us who pay close attention to this illness will read some new development almost weekly. And it's very difficult to ascertain, what's important, what's less important. And then also, how does it all fit together? Well, I can tell you that Dr. Vernon has a very orderly mind. And because she's been focused on MECF research for 2 decades, she really can help us put it together in ways that make sense for us, and that's what she will attempt to do today. So it's a treat to have her summarize the 2014 research for the rest of us who are not scientists. So with that, Suzanne.
Thanks, Carol. So our goals for today's webinar are to, first of all, I'll take the first 10 or 15 slides to really distill some of the recent and really promising, exciting areas of research. Then Carol will come back in and she'll give us an update on what's happening at the federal level. And I hope by the time we're finished that we will have helped you understand how important a lot of the attention that's being given to MECFS, as well as the incredible research that's going on, the importance how important that is and the impact that it actually has on our community. So if we think back, and actually this slide comes from when MECFS was really put on the federal radar in a way like never before, when the FDA went on record as saying that MECFS is a serious condition with unmet medical need. Now, for a federal agency to do that, particularly the federal agency that regulates the way drugs are developed for a particular disorder, and in our case, MECFS, that is just highly, highly significant. It puts MECFS on a radar in a way like it has never been done before. So when the FDA does this, the FDA defines MECFS as a serious condition, and by their definition, that means that it is a condition that has significant impact and morbidity on a day-to-day basis and affects the way an individual lives and functions. unmet medical need, as defined by the FDA, is when a disease has no treatment or it has inadequate diagnosis, and therefore it translates to an immediate need that needs to be addressed at a number of different levels from the public and private sectors altogether. So, the reasons that MECFS is considered to be serious is because, number one, it's incredibly debilitating. It's highly prevalent. There are far too few clinicians who understand it, who diagnose it, and who treat it. And as I indicated above, it's significant unmet medical need because there are no approved diagnostic tests or treatments. So that makes the research that I'm going to tell you about in the next 20 or 30 minutes or so all the more important, because with the type of research that's going on, we do it in a framework that allows the FDA and the other federal agencies to understand how it's contributing to the evidence base. And again, this is an evidence base that is needed in order for us to ultimately come up with the objective diagnostic tests and the treatments that are required to really address the unmet need. So this is a piece that actually came out of our winter issue from the Solve CFS Chronicle in 2014. And Dr. Tony Komaroff of Harvard Medical School, who many of you may be familiar with, and I kind of put our heads together and came up with what we believed are some of the top 10 most important recent research advances, I would say over the past 4 to 5 years, how they've really come to fruition. Anyone that's noted on this slide with a star is one that we've actually had a hand in a role in supporting and contributing either dollars or samples to the biobank. You can read more about this actual article, if you go to our website down here at solvecfs.org, the Research Digest 2014 Top 10. What I'm going to do is I'll talk a little bit about all of these: delineating heterogeneity, oxidative stress, inflammation in the brain, central sensitivity, autoimmunity, patient-centered research. This is kind of a relatively new phrase that in many ways our organization is very much spearheading for this community. Risk factors, brain white matter, the autonomic nervous system, and pathogens, and how many of these research areas are now beginning to not merge, but really when thought about together, how one is influencing the other and impacting the way that we actually think about MECFS and the ways that we're beginning to further study it. So in the next couple of slides, what I've done is I've grouped these together and to illustrate further how the research is, there's a lot of synergy in the research that's being done by us and by others, and how this is really beginning to connect the dots in many ways for research. So let's look at heterogeneity and patient-reported outcomes. Over here on the left-hand side of your screen, we now have substantial evidence that MECFS is quite heterogeneous. And what that means is that one individual's disease is not necessarily like the others, and that's reflected by these different colored little people here in this picture on the right. What we've been able to do using ways of getting people to report on how their disease affects them, and also by using molecular tests like gene expression profiling and genetics, we've been able to now begin to identify, sort people, delineate this heterogeneity, if you will, into very more homogeneous subtypes. The reason that this is important is because it increases the likelihood that we're all studying the same thing, if you will. So when we're able to say subtype 1 is associated with a specific type of biomarker, let's say a virus, subtype 2 is associated with another type of biomarker, let's say a brain abnormality, etc., that gives us the opportunity to the subtype and increasing the likelihood that we will identify a biomarker or biomarkers associated with this specific subtype. Again, extremely important for getting to an objective diagnosis approach, as well as for focusing in on treatments. This wouldn't be possible, this type of subtyping wouldn't be possible were it not for what is called patient-reported outcomes. Patient-reported outcomes is the way that an individual experiences their disease, and in our case, experiences their MECFS, and how that, that disease is affecting the individual. So an outcome is the way a person feels after a particular treatment, in response to a treatment, in response to diagnosis. There's several efforts actually going on that are looking at patient-reported outcomes, specifically the CDC multi-site study, and some of the other NIH-funded studies, and we've also been using our Solve CFS Biobank to get a handle on patient-reported outcomes. In a study that we did in response to the FDA meeting last year, we were able to show that, in fact, many of the outcomes that patients report as being important to them are assessed in the research studies and are reported in the literature, but then there's this list that I show here of things that patients have reported as being very meaningful to them, but yet we have not been measuring them or researching them adequately. Things like vertigo, things like difficulties in breathing, things like changes in taste, and various types of pain and sleep associations. So again, this is important because it gives us clues for types of directions of research that we might begin to look into further. Oxidative stress and inflamed brain I've put together because one actually can cause the other. Inflammation results in oxidative stress. Oxidative stress can also further inflame inflammation. This is actually work too. The oxidative stress picture that I show here on the left is a picture of a brain, and the red hatched marks are called the ventricular spaces of the brain, and this space is the space that's filled with cerebral spinal fluid. We supported investigators who now have gone on to receive NIH funding to understand why there is increased oxidative stress as measured by increased lactate in the brain of CFS patients. And they are now actually funded by the NIH to look at ways to treat that oxidative stress using supplements and other treatments to bring that down. Another very important finding that was just published this year was evidence of neuroinflammation. This picture is shown on the right. This is actually a study that was published in the Journal of Nuclear Medicine by the Japanese group, Professor Watanabe and Hirohito and colleagues at Osaka University. And what they were able to show is that the immune cells in the brain, these immune cells, one type of immune cell in the brain is called a microglial cell, that these microglial cells were highly activated. And when a microglial cell is activated, as shown here by this yellow pattern in this picture of the brain, that means there's inflammation going on. So this is evidence of neuroinflammation in the brain of MECFS patients. This was actually the first demonstration of neuroinflammation in the brain of CFS patients, and I have communicated with Professor Watanabe recently, and he just told me that they've finished testing an additional 10 patient-safe controls. The study that was published actually looked at 9 CFS patients and 10 controls. And there are additional studies, both in Japan and in Sweden at the Karolinska Institute, has continued to show this type of significant finding of increased neuroinflammation in the brain. Now, that's very important, again, from a diagnostic perspective, and also a clue of the types of things that we can begin to look for in the blood that helps us understand further how to diagnose neuroinflammation, for example, using a blood test in addition to something as a brain image.
Suzanne?
Again, importantly, oxidative stress and neuroinflammation are related. Yes.
Quick question. Someone is asking if more neurosurgeons or neuroradiologists that specialize in this type of MRIs are we looking to pull more of them into research? Do you see more of them coming into the research field now that we're starting to see this inflammation on the brain?
That's a very good question. I should find out who Professor Watanabe is working with at Karolinska. I believe that he does work with the neurology group at Karolinska. I know there are a number of neuroscientists that are now interested in CFS, including Patrick McGowan, who we work with. And I also believe that there is the neurology group at Harvard that is doing something similar to try to further validate the Japanese findings of neuroinflammation. So this is a super hot field right now, so I'm certain it will continue to attract the interests of people that are expert in neuroimaging. I grouped the autonomic nervous system and central sensitization together because they're both connected, and because I think that one is some of the findings as far as autonomic nervous system and orthostatic instability and postural orthostatic tachycardia syndrome, which we know is very common in MECFS patients, how that may be affected by this phenomena called central sensitization. So on the left, I'm sure many of you are familiar with this particular setup. It's a tilt table, and it's used frequently, or it's used in MECFS patients to really test how well blood flows through the body. In MECFS patients, there's this phenomena of blood, for whatever reason, pooling below the belly button, and for whatever reason, not being able to flow adequately up to the brain. So that when a patient is tilted up, as shown in this picture, the blood does not get up to the brain adequately, and it can cause fainting. And in research that we funded, as well as research that's being done at Vanderbilt University, at the Mayo Clinic, and many other academic settings, what is really being shown is that these autonomic nervous system abnormalities are quite common, and now there is an investigation as to ways to overcome this inability of the blood to flow adequately through the body. With these autonomic nervous system abnormalities comes an exacerbation or a worsening of symptoms. So a worsening of brain fog, and for those of you who have actually experienced this tilt table phenomena, this can induce a significant worsening of many of your symptoms. And it's now thought that central sensitization may actually have something to do with this worsening of symptoms that occurs under a number of circumstances. And many of you may be familiar with central sensitization in fibromyalgia, where we know that the signals in the brain are amplified. So if you think of it this way, pain, for example, a simple touch on the skin should not cause pain, but in people with fibromyalgia or chronic widespread pain, that touching sensation, the signal is amplified dramatically in the brain, causing a simple touch to actually be felt as severe pain. And this is what that central sensitization phenomena looks like in pictures of the brain. This top panel shows kind of brain slices. This is an imaging of certain types of markers of activation in the brain. So this is normal transition. So this is someone who does not have central sensitization and is being touched, for example. This is what a normal pattern would look like. So normal axon or synaptic transmission. And then below is the exact same phenomena in a brain from a person with central sensitization. So you can see all of these signals are significantly amplified. And again, this is what's thought is occurring now also in MECFS, where not only are signals that transmit pain sensations to the brain amplified, but also central sensitization may in fact be accounting for brain fog amplification, this inability to concentrate and think adequately, as well as actually fatigue. And Peter Rowe at Johns Hopkins, who's doing a neuromuscular strain study, has shown that even the simple lifting of a leg while the individual is laying on their back and the leg is actually being assisted can induce severe fatigue and pain because of, or potentially due to, this phenomenon of central sensitization. I grouped together autoimmunity and risk factors because we know that many autoimmune disorders, diseases, are much more prevalent in women. And that's why I have this woman shown here at the bottom right. We also know that MECFS occurs much more frequently in women. And most studies have shown that women are at 2 to 4-fold increased risk for developing MECFS. In work supported by our organization, we are showing, and we will publish soon, evidence for an autoimmune subtype in MECFS, and this is work that was done using the Solved MECFS Biobank. And as described in the Chronicle, there are a number of intriguing studies that have been published, as well as trials that have been conducted that are implicating autoimmunity as a role in MECFS, particularly in potentially subtypes of MECFS. And this is further exemplified by the rituximab trial that was conducted in Norway, where they used a drug, rituximab, that takes out B cells. B cells are the cells of the immune system that produce antibodies, and those antibodies can attack self, so that's what autoimmunity is about. And when these MECFS patients were treated with rituximab, a portion of them responded quite well to treatment, again, further implicating the possibility of this autoimmune subtype. So autoimmunity appears to play a role, at least in a subset of MECFS, and that's important from a treatment perspective as well as potentially a diagnostic perspective, because that could give us the chance, the opportunity to use biomarkers, or I'm sorry, antibodies or patterns of antibodies, these autoantibodies, as biomarkers to diagnose these subsets. This picture down here is just, I borrowed this from one of our webinar presenters, Dr. Nuryat Ritmaz, who demonstrated that the immune system, when in balance, does a great job of clearing pathogens, repairing damage, but when it gets out of kilter, it can actually contribute to inflammation and autoreactivity. And again, this is probably a very important subtype for MECFS, given that we know women are at increased risk and some of the other risk factors, including allostatic load, which is an indicator of long-term inflammation that has also been shown to be increased in MECFS patients. Pathogens
Suzanne, sorry, we do have a question.
Together.
Suzanne.
Yes.
Sorry to interrupt, but we had a question on the previous slide about is autoimmunity tied to women's hormonal presence, so there's less emphasis as the woman enters menopause? Is there any connection between menopause and autoimmunity?
I don't know if I know the answer to that. You know, I do know that our immune system is regulated by a number of hormones, particularly estrogen. But I'm not sure I feel that I know enough to say anything more than that.
Okay.
I'm sorry. So I grouped pathogens and white matter together because, not because this particular finding of late, which was the white matter finding, is that we know that it's caused by possible pathogens in the brain, but you know, it is plausible to think that perhaps the pathogens or the viruses and the bacteria that we live with on a day-to-day basis could in fact be affecting these findings, these imaging findings that were shown in the white matter recently. But first, let's start on the left with pathogens. Our organization has actually funded some of the first virus work and has continued to fund research that explores the role of various types of pathogens, viruses and bacteria and parasites, and their role in MECFS. What Dr. Komroff and I thought was particularly important was this most recent research that really tries to understand the effect of latent viruses. Again, viruses that kind of stay dormant and viruses that all of us have, and why they cause damage in people with MECFS, and yet, you know, healthy people that have these same viruses, they don't cause any damage to. And we're really beginning to, I think, appreciate this imbalance that may in fact have something to do with the immune dysfunction that that we know exists in MECFS patients, and perhaps some component of genetic vulnerability for whatever reason, the MECFS patients are not able to keep these viruses in check, and also the connection between infection, particularly with viruses, and the production or the generation of an autoimmune response. One of the things that we know is that viruses have proteins, and this is a picture, a really cool picture that I borrowed from Vincent Roccagnolo of influenza virus. But we know that viruses have proteins that are very similar to proteins that we have in our own body. So that when we are generating an immune response to clear a virus, such as influenza, in turn we can also be generating some, that same immune response can also begin to attack, if you will, parts of our own bodies. So we're just beginning to understand this, but this is a very important area of research. We also know, are beginning to appreciate much more, that viruses that infect one part of the body, for example, here's a picture of flu, and flu we know replicates in the respiratory tract, how flu infection, acute infection with influenza, can actually cause severe gastrointestinal symptoms and signs, even though the virus is never there. So again, this idea of how the immune response is really responsible for orchestrating, connecting, and regulating a lot of what goes on in the body, both good and sometimes not good. This picture on the right is a picture of the arcuate fasciculus, which is a nerve bundle shown here in these red kind of lines or cables that connects two areas of the brain that are important in signal transmission and language. These are, this particular bundle connects these two areas of the brain that are really important in how speech and language is processed. And there was a study just published by Stanford University from Dr. Jose Montoya's team that showed that this particular area of the brain, the arcuate fasciculus, is increased in MECFS patients, significantly increased. Again, this is a very novel finding. We don't know what it's caused from, but it could be associated with the inflammation that I've shown you we now know and appreciate occurs in MECFS patients, as well as the inability for MECFS patients to regulate the types of viruses that we all live with on a day-to-day basis, but in MECFS patients wreak havoc. So that is a very quick overview of an enormous amount of research that's going on. And I would say an enormous amount of very exciting research that's going on in MECFS research. And I think you can see how a lot of this research that's going on in these very different areas, and in many cases by very many different investigators, many that we funded and many that we collaborate with and many that are working independently, how this information is beginning to converge into a picture that's really, I think, giving us an understanding much more of the disease process that's going on in MECFS patients. Over the past year, we've been talking to a lot of investigators and really trying to understand how to make the research process for MECFS much better, much more efficient, and accelerate the process that we have from the basic research, these basic and very important discoveries that I've just told you about, to getting these things into the hands of physicians in order to be able to objectively diagnose and in order to be able to treat. So where the conversations that we've had is really to understand where are the bottlenecks in this process. How can we have all of this cool research, but yet we still don't have any treatments, any objective diagnostic measures? And time again, what we've heard is that the bottlenecks in the process are, number one, engaging enough patients, well-characterized patients, into the process of research so that we can have enough individuals in our research study, have enough individuals in our clinical trials, and then have the ability to actually validate these studies or the findings in an independent population, for example. So two very clear bottlenecks: engaging enough people in the research process, and then being able to have a resource, a population, a biobank, a registry in order to be able to validate those findings on this end. So very important bottlenecks that we've, over the past couple of years also, have recognized and have built our Research Institute Without Walls with our Solve CFS Biobank right at the center in order to be able to address those. And this is what we're really going to put into play, not only next year, but for the next several years. We are building our community in a way that allows us to engage a very diverse population of people affected with MECFS, as well as people, family and friends, and people who want to participate in the research process. Okay? Engage them using tools like the internet, using tools like specific apps that allow us to allow the individual to be able to record and report what is important to them and to their disease and well-being. So this is our really phase one, engaging a broad and representative community to participate in research and making this a kind of a sticky process, a process that translates into benefit for the individual. In other words, having access to the most recent and important data or information from what the others in the community say that you can actually bring to your doctor, and your doctor sees this data and is able to actually take this and use it in your care and the care of other MECFS patients. We engage this community to provide information, data, samples that allows us to now further dive deep into describing what MECFS is at a molecular level. So with samples we can generate genomic types of data. We can do microbiome and virome types of data. Again, in each case, making sure that all of this molecular information is linked to the individual. This is a very patient-centric, a very unique approach to describing and ultimately defining MECFS at a molecular level. This will allow us to really identify MECFS subtypes in a way that hasn't been done, mostly because this is big data like we've never had, and hopefully we'll soon very much see in 2015. Importantly, all of this data that gets generated by the individual and connected along with all of this molecular information will be shared, will be put into a data repository that can then be accessed in a secure way so that we can have the brightest minds looking at it, analyzing it, combining it in a way that has not been done before. So fresh eyes, bright minds, incredible computational power to put all this information together to not only define subtypes, but ultimately to get us to this place where we have objective diagnostics and we have a really good idea of some of the potential targets that we can identify and target for treatment. And we announced this last night, but we got an incredible jump start on our 2015 plan, and that was a $500,000 award from the Falk Medical Research Trust. This was a funding opportunity that we applied to because we thought it fit us perfectly. They were really looking to fund organizations or academics or universities that focused on treatments and cures for diseases for which there are none. And so we fit perfectly. And we have the honor and the privilege to be able to partner with Drs. Lucinda Bateman, who will be our clinical collaborator on this award, and Dr. Patrick McGowan, neuroscientist up at the University of Toronto, who's going to be doing the epigenetic piece of this study. We have 1 year to be able to do what we propose to do in this. In addition to the epigenetics work, what this study will give us the opportunity to do is restock our Solve CFS Biobank inventory. So we'll be going into 2015 and the years beyond with, again, building our Solve CFS Biobank to be a very robust resource that continues to attract investigators into MECFS research. And I would also like to say that we have already, we will be collaborating with investigators like the Japanese team, Professor Watanabe, to explore these samples for markers of the neuroinflammation in the brain that they have identified. This is just so exciting because it was an incredibly competitive process, and I think it was really just a validation and affirmation of what the Solve MECFS initiative has been able to accomplish over the past several years with the help of the phenomenal support of all of our patient community and our donors. So without further ado, we'll transition now to Carol, who will give us an update on what's happening at the federal agency level.
Well, thank you so much, Suzanne, for that summary of a large body of information and a complex body of information. I appreciate it very much. I'm going to talk a bit quickly through this because I do hope we can allow for time for questions at the end of this webinar. So let me answer first the question, why are we talking about the work at the federal level? To go back, our organization really is focused on facilitating research, and we do that in a couple ways. One is we directly fund research that we think is the most promising. Second, we facilitate research that we don't fund with samples from our biobank and patient registry. And last, we continue to advocate for additional funding from the federal government. For most illnesses, the federal government provides the strongest source of funding, and we certainly know that that has not been the case in our illness. So as a coda to Suzanne's presentation, I want to talk a little bit about the federal. To be clear, we don't involve ourselves with the federal as an advocacy organization. We understand that there are many points of view in the patient community about any issue you could name. However, we feel it's important to bring the scientific knowledge that we possess, you've just heard Suzanne describe it, to the federal setting so that there is rigor to the discussion about the illness on the federal level. So with that, we bring attention to the illness primarily through the CFSAC. And the CFSAC, again, is the CFS Advisory Committee, and we are quite fortunate to literally have a seat at the table on that committee. It's the primary long-standing committee of Health and Human Services that makes recommendations regarding our illness directly to the Secretary. And we are pleased to be on that committee. We also, like many, are frustrated by the very, very slow pace of change. And at the same time, we feel it's better to participate in it, to be the one who can bring the rigor of the scientific approach to solving this illness to federal discussions, so we stay with it. Next slide, please. So just an update on recent developments. First, last year, 2013, in the spring, was the FDA Voice of the Patient Report, which really was a landmark discussion about our illness. And brought together many experts and shone a light on the illness that had not been so before. The next up is this Institute of Medicine study, which was contracted by Health and Human Services to make recommendations regarding our illness. And that report will be published in the spring. We're now just months away from a long-anticipated and by some long-feared report. There are 4 very specific charges for that esteemed committee that makes up the Institute of Medicine. One is to scan the research, and of course, as a research institution, we're delighted that all their thinking and work starts and ends with the research. Second, to recommend a new name for our illness. Third, to clarify a new case definition for the illness. And this is not an academic issue. Much of research has been held back because of the variety of case definitions that are in play. So this is a very important recommendation for the Institute of Medicine Committee. And last, to develop a specific plan to train United States medical personnel, doctors, nurses, and so forth, regarding this illness. So again, those 4 charters are very important. The third effort listed here is the NIH Pathways to Prevention, which the primary workshop related to this was held just last week. Suzanne, Dr. Vernon, was a speaker at that event, and the goal there is to fill the gaps in research into MECFS. And much as we had the opportunity just now to listen to a rich discussion of the research into this illness, I think as we've seen, we are still at the place of doing basic and translational research, and frankly there are as many gaps as there are answers. So we do await with some trepidation the P2P report, which again will be published this spring. And then last is the CDC-sponsored multi-site study. This is a study that began in 2012 at 5 sites in the United States with 450 patients. And you know, we are glad to see research into the illness, and obviously the jury is out on what will be found there. So that was a very quick run through the federal efforts. I would ask you, as I wrap up here, just a couple of notes before we have some time for a couple of questions. If you felt that this was a useful session for you regarding the work of our organization and the research, please do consider signing up for the newsletter. When you sign up for that, you will get regular updates on the research with our take on what works, what doesn't work, and what it means for patients. Second is joining the biobank. We urge everyone, patients and healthy people, to become part of our biobank. It has become a critical resource, and we plan to expand it significantly in 2015 to help fuel the project that Suzanne described. And last, you know, none of this can be done without dollars, and we are grateful for every donation. We were thrilled to get the FALK grant of $500,000 just last week, and at the same time, that money takes us forward in being able to do research we couldn't do otherwise, but there is so much more research to be done. Our donors have been a long-standing bedrock of what has fueled our organization. So I'll tell the many on the webinar, if you've donated to our organization in the past, I hope you feel proud that you have helped to pay for the research that Suzanne's described. And we urge all of you to move forward and consider donating to us. We currently have a matching grant from one of our very generous donors, so any donations through end of the year will be doubled. So with that, thank you very much. If you have any questions, and we have a couple minutes, you should type them into the box on the lower right-hand side. And we really are very, very grateful that you've been with us through this session. So I will hand it over to Lee then to bring forward any questions.
Great. We do have a few, some that go back, of course, to what Suzanne was talking about. One is a question about for all these results that are coming forth about neural inflammation and other brain inflammation, are they including in their study patients that do not experience brain fog? Do you know anything about those specifics of those studies?
For the Japanese team, the eligibility criteria were CFS patients, and they actually, and defined by the 1994 Fukuda criteria, and they correlated inflammation to symptoms of pain, brain fog or cognitive impairment, and fatigue. And the interesting aspect is that they were able to show that different areas of the brain were inflamed according to kind of the severity of the particular symptom in the individuals. Now, again, these are relatively small studies, but despite how small they were, very significant, and inflammation correlated with the specific symptoms, including cognitive impairment or brain fog.
Great. We have a question about a study Dr. Komarov participated in 1995 on disequilibrium. I'm wondering if there's any new research regarding this this symptom with CFS.
Mm-hmm. No, but I don't know, and I confess I'd have to go back and look at that study. But this term disequilibrium is very interesting because in the work that I presented last week, as we've been analyzing the patient voice in response to the FDA survey, this idea of disequilibrium as being a symptom that's frequently experienced in MECFS population and our community was quite high. And again, it's one of these symptoms that we haven't paid enough attention to in research, and so yeah, it's
okay, great.
Definitely worth looking into.
We are getting a few questions about the biobank. Wow, a whole bunch just popped in, sorry. We're getting a few questions about the biobank and how their results are tied to them, and if they have access to any of the results once they offer a donation of blood that's used in the study, sort of what that feedback loop is and how it's kept confidential.
So when an individual participates in the biobank, you do not receive individual results. And the only time that happens is if for example, in a particular study, and the testing criteria, the inclusion criteria or exclusion criteria is, let's say, for example, HIV. You cannot be HIV positive and participate in this study. And so in order to do that, the investigator tests for that. And if an individual comes up positive, then that is something that will be reported back to the participant. and then the participant takes that information to their physician. Otherwise, it is all research information, and we provide results back in the form of kind of summaries of the research data in our Research First blog posts, our research digests, and in the publications that the investigators report. So you have to think of your participation as part of the bigger picture, part of solving this puzzle. Yeah. So no individual reporting back.
We are getting some questions about qualification of patients in the biobank, and how do we ensure the folks actually have chronic fatigue syndrome, so forth. So could you talk a little bit more about how we qualify individuals within the biobank for studies?
So we actually cast a broad net. We take many people who want to participate in the biobank, and then by collecting specific types of information, using specific types of questionnaires, and conducting kind of standard blood panels to exclude certain medical conditions is how we qualify individuals. And the questionnaires are based on both the Fukuda and the Canadian criteria, and it also involves whether or not individuals are taking certain types of medications. It involves how old individuals are. So again, study criteria for specific types of studies varies. But in general, to qualify as an MECFS patient, you would have to respond appropriately or to the various questionnaires in order to qualify.
Great, and we are actually at the top of the hour, so I'm going to go ahead and stop the recording and thank everyone very much for participating. We do have quite a number of additional questions, so Suzanne, how would we like to handle those?
I would love to see the questions, and we can respond to them in a blog post. How's that sound?
Sounds great. So everyone can look for that. Know that the recording of this webinar will be made available online by the end of the week, as well as the slides. And as Dr. Vernon just told you, we'll get to as many questions as we can in a blog post as well. So I apologize if I was unable to get to your question. There were a lot of them. A lot of them came flying in at the end. So we'll do our best to get back to you in the coming weeks through our blog.